The third and the last of the processes in the central dogma of molecular biology is translation, or protein synthesis. Alright, so again, from the name itself, we could see that the process here, or the end point, is the production of proteins, but its source again is RNA, particularly mRNA. So we could also name translation as RNA directed protein synthesis. And we know that in protein synthesis we use the ribosome, which is in the cytosol but outside the nucleus, to produce the proteins out of the mRNA that we have produced. Alright, so first things first, we should be able to identify which of which things we need for protein synthesis, and I've already listed them down here. First, of course, we have the gene, or the mRNA, that has undergone transcription in post-transcriptional processes. Second is, of course, the ribosome. Of course, without the ribosome, we wouldn't have the organelle which would do the protein synthesis. Alright? The third would be tRNA. Alright, so we have here another type of RNA, but it is tRNA. T stands for transfer. The purpose of tRNA, very much unlike the mRNA, is to be able to bring the amino acids to the ribosome. And these amino acids are actually the components that will make up the proteins that correspond to the gene, alright, to whatever the ribosome would read of the gene that comes into it. Alright? And 5, a bunch of enzymes that that we will discuss later on. First things first. Alright, so we have amino acids here, and the amino acids that the tRNA will bring are going to correspond to a particular code in the gene. And the name of that pattern in the gene which will code for one amino acid is a codon. Alright, so this is located in another slide, in another paper, but I'll mention this already. A codon is composed of 3 nucleotide bases of the mRNA, and there's a particular anticodon in the tRNA that would read that codon. And depending on the anticodon, alright, whichever codon would correlate to a particular anticodon will bring about the amino acid that the tRNA would bring. That means that if the codon here was different, then the anticodon would also be different, then it would also correspond to another amino acid. So this means that the amino acid that the tRNA would bring with it is dependent on the anticodon it would read, which is dependent also on the codon that will come out from the mRNA that will undergo the translation process. So before that, we should be able to know that the amino acid must attach first to the tRNA, because in the start they are separate. So the tRNA here must be able to combine with the amino acid. So the process of that is known as activation. So the tRNA, when bonded already to the amino acid, is called an activated tRNA. Alright? So how does that happen? So there's actually participation of a particular enzyme. Alright? And the name of the enzyme here is aminoacyl tRNA synthetase. So what it's going to do is that it will combine the tRNA and the amino acid. So now this one is called an activated tRNA. Alright. Now, another thing I would like to mention is the ribosome here. Alright? In reality, the ribosome is actually divided into 2 parts. One part is larger than the other, and they are given a particular number, and that number is going to end with an S. Alright? For example, in the humans, or in eukaryotes, we have the 80S ribosomes, alright, in eukaryotes. And in prokaryotes, you have the 70S. Alright. For example, this one is the 80S. The 80S is actually composed of a 60S subunit and a 40S subunit. While for the 70S, we have a 50s and a 30s subunit. So you would see there that this is not, the 80 here is not actually the sum of 60 and 40, as well as 70 not being the sum of 50 and 30. Alright, because it's not really like that addition. The S here stands for Svedberg. It's a unit of sedimentation. Alright, so it designates how long, or the degree of settling or sedimentation of the ribosome, alright, based on centrifugation. Now, for example, we have here the 60S and the 40S subunit. There's a particular role of these two subunits, as we will see later on. They are separate at first, and they will join when translation is taking place. Alright, so with that said, I have to note that there are several properties of translation, alright, specifically for the mRNA, or the gene that is being read. Alright, the first of these properties is non-ambiguous. Alright, we know that ambiguous stands for confusing. So translation should not be confusing, meaning that a particular set of 3 nucleotides, which is basically a codon will only code for only one amino acid. Alright, for example, I have the codon AUG. This will encode particularly methionine only. There will never come a point wherein the AUG will stand for another amino acid. As long as you have AUG, it will always code only for methionine. Second, degenerate. But if you put the non-ambiguous rule the other way around, the codon will code only for one amino acid, but for an amino acid, one amino acid could actually code for, could actually be coded by more than just one codon. All right. I'll try to Google one. I'll try to look one, for example. All right. For example, in my reference, I have histidine. It can be coded by CAC or CAU, meaning that we follow the non-ambiguous rule. CAC will always code for histidine, but so will CAU. But this just means that for histidine, this one could be coded for by 2 different sets of nucleotides. So that's the degenerate rule. Okay. The third is non-overlapping. And might as well include the fourth, which is commaless. So 3 and 4 are kind of joined together. So in non-overlapping, it states that a nucleotide can only code for one amino acid. What do I mean? If I have this codon here, I have the first, the second, and the third nucleotide of this codon. The third nucleotide cannot anymore be part of the next codons. So if I have this one, the third nucleotide, this one cannot anymore be part of something like 1, 2, 3, then 1, 2, 3. No. If I end with 1, 2, 3, then this one would be 1. This one cannot be part of the next codon, so this one shouldn't be overlapped. As well as commaless. So if here we're talking about overlapping, here we're talking about gaps. So for example, I have 1, 2, 3. I cannot start with 1, skipping this nucleotide. I must go from 1, 2, 3, then this is 1. You cannot skip that, or you cannot put a comma on the code, because this one should be read straight. Alright, that's why this one is also termed as no punctuation. All the nucleotides must be read. You cannot skip. And then finally, universal. Meaning that for humans, if I have AUG coding for methionine, most likely in fungi and plants and in bacteria, somehow, AUG will also code for methionine. It's the same throughout all species or organisms or even kingdoms. Alright. So here, I also mentioned this one cannot be part of the third. That's the non-overlapping rule. But if I talked about fidelity of replication and fidelity of transcription before, there's also a kind of fidelity here, meaning that there's actually a possibility that the codon Alright, or the tRNA will give the wrong amino acid for the codon that is being read. So that's kind of the same thing as with before, alright? There's a kind of mistake that can come up even if the codons were correct. That's why there's a property of tRNA, right? They have wobble bases, alright? Meaning that for the third nucleotide of the codon in the anticodon, in case that there is a mistake, this would kind of be, the third one would kind of be loose, alright? It will not be very tight so as to give way for corrections in case, for example, tRNA gives the wrong amino acid. Hey, I think I read you wrong, codon. Maybe my wobble base could loosen up and I'll give up the proper amino acid instead of the wrong one I initially put. So that's wobble base, kind of wobble, because when it's loose, it wobbles, you know, or gives that kind of wiggling motion. Wobble. Anyway, enough of the introduction. In translation, we also have the same phases. Of course, we have the initiation phase, the elongation phase, the termination, and some post-translational processes, but not really in any way similar to the post-transcriptional processes. So let's start off with protein synthesis with initiation. Alright, with initiation, all we do is we open up the ribosome and we insert the mRNA there. Right, we just set up the entire apparatus, if I would call it that way, in order for elongation to take place. So in order for that to happen, alright, the first thing we have to go to is the formation of a particular complex with this one. As I said, the ribosome is composed of two separate subunits, and in initiation, they have to be joined together. Alright? So, in order for that to happen, we have several what we call initiation factors in order to set up the entire apparatus. Okay? So, first thing, the initiation factor 1 assures that the two subunits are separated. Alright? They are separated. Alright? So, I stand corrected. They're actually joined initially, but they are separated, but then joined together later on. Alright? Then, we have mRNA. Alright? We have the mRNA here, and the mRNA are actually linked with the lighter subunit, and in correlation to that, the tRNA are joined with the heavier subunit. So if I have here the 50S and 30S subunits, mRNA are supposed to combine with the 30S. Alright, so we're talking about initiation in prokaryotes here because we have 50 and 30S instead of 60 and 40. And for the tRNA, they would be combining with the 50S subunit. Alright, so for that to happen, initiation factor 3 would actually form the 30S complex, and that means the 30S subunit plus the mRNA. So IF3 is responsible for this one, and the one responsible for this one, okay. I mean, this one is not really this one is not really initiated by any factor because the tRNA come and go. But the thing here is that the 30S complex would now combine with 50S. Alright, so 30S plus the 50S subunit will yield the 80S complex, and that is initiated by initiation factor 2. So again, mRNA would now be combined with the 30S with the help of initiation factor 2, and then 50S will combine with the 30S complex using initiation factor 3. And now that you have the mRNA sandwiched between the two subunits after combination with the initiation factor 3, the tRNA here will now start doing the elongation process. So the elongation process would start by having the first tRNA activated that is. So let's put amino acids to denote that it's activated. And the first tRNA will go to the P, alright, to the P portion, and it will now read for the first codon. So for example, okay, it will now read for that, and it now gives us the first amino acid. So it's initiated. We now go to the elongation portion. So as I said, the first tRNA will go to the P area, and once it is confirmed that the codon here is correct, the amino acid is correct here, the next tRNA will go to the A portion. Alright? So the A portion will then do the same, read the codon here, and then let's assume that this one is read correctly. Okay? So what's going to happen here is that when these are both correct, there would already be, remember, these are two amino acids. Now, there will be a peptide bond formed between this, alright, using a particular enzyme. And the name of the enzyme is peptidyl transferase. Alright? Peptidyl transferase. So, here, the first and the second amino acids are combined. Okay? And after this, now you have a dipeptide. The next thing we have to know is that this one is trimmed. Alright? So what's going to happen is that the second and the first amino acids will now go to the tRNA at the A area. Alright? At the A. So the tRNA at the P area will now be removed of the amino acid it usually had, which is amino acid number 1 here. And the next thing that's going to happen is that the one at P will move to E, the one at A will move to P. So what's going to happen? 1 is at E, 2 is at P, A is not anymore occupied. But the second tRNA will now hold amino acids 2 and 1. Alright? Again, this one will hold 2 and 1. Okay? So, since A here moved to this one, 3 is now free to go to A. So, what's going to happen is it will be the same again. So, what's going to happen? A peptide bond will be formed. This one will be trimmed. So, we see that as we move on with the addition of the new tRNAs, the latest amino acid will be the one which is going to be held by the tRNA currently at the A area. So this will go on and on and on until we reach, let's say, like 1,000 peptides. I mean, 1,000 amino acids maybe. So let's say we reached 1,000 amino acids. So we now go to termination. Again, I want to review you guys. So for example, this is the 1,000 tRNA. It's supposed to be, there should be the 1,001st to go here, and then which will bond to the 1,000th amino acid. But in case of termination, the one that will go to the A area now is not anymore a tRNA, but a certain RF, or a release factor. So since the release factor here does not have an amino acid, what it's going to do is that it's going to go here. So we have the release factor at the A area. Alright, originally what's going to happen is that there would be a peptide bond between this one and the amino acid here. But since the release factor doesn't have any of that, what it's going to do is to act like a scissor, wherein it will do what usually happens in the P area. Again, is there's a cutting part here, but here it's going to happen also, but this time the protein synthesis will stop because there is no more additional amino acid. So this means that this one is already the last amino acid in the peptide chain being formed. And so since you have that long peptide there, alright, and you cut that out, the protein strand or the polypeptide strand stops there, and boom, termination occurs. So just like what the RF means, it releases the polypeptide chain from the initiation complex, or from the ATS complex. And we finally have here 1,000 amino acid containing peptide. So the thing here is, let's say it's already released. Now we have a free polypeptide. And that's, of course, the product of protein synthesis. Alright, we got what we want, but just like in transcription, the polypeptide is not really functional. Remember, proteins have their levels of organization. The level of the polypeptide we got here is primary only. And it's primary, yes, because remember, in the primary level of organization, we need peptide bonds. And what we did here in the elongation part is to put peptide bonds one amino acid after the other. So here, when we produce, after termination, when the release factor has cut off the polypeptide from the ATS complex or whatever complex it is, depending on the ribosome, the polypeptide here is only the primary level of organization. But we know that many proteins are functional not just in the primary level, but instead in the secondary or even greater levels of organization. So there has to be post-transcriptional, post-translational processes. And the first, of course, is folding. They fold, they have their hydrogen bonds or their residue interactions to get to the level of organization they want in order to get to their native conformation, or the conformation wherein they do their thing. Alright, because if it doesn't fold like that, it will not really function at all. Another thing that can be done is modification. Alright, modification. So modification is done, okay, in order for them to be even more active. For example, trimming away. For example, there are really some amino acids in the polypeptide that was formed that's not really going to be used. Alright? Or even residue modification, meaning that, for example, I have proline. Modification would be a set of chemical reactions. For example, in proline, I could put a hydroxyl group here to get hydroxyproline. Something like that. So you get a derived residue out of the 20 residues we originally have. So many proteins are like that. They have special residues that are not really part of the 20 amino acids, but they need those modifications in order for them to work properly. And also, we could also add prosthetic groups or coenzymes in case the protein that you are synthesizing is actually an enzyme. So you add metals, you add cofactors such as the vitamins in order for the protein you just produced to work properly. Alright? So that those are in cases that you got a good polypeptide and you want to modify them in order for them to work. But what if the protein is not really the one that you got? It got away from, it was coded for by the right mRNA, all the codons were correct, but the amino acids were just wrong. The protein was wrong as a whole and it has to be destroyed. Well, there is also a path to degradation. Alright, there are several. For example, the one I have in my reference is basically we have the term proteases or proteasomes to degrade these erring polypeptide chains. Alright, and a pathway, for example, a particular biochemical pathway is the ubiquitin pathway. And then finally, protein targeting. You want some of these proteins to go in certain parts of the cell. So the Golgi apparatus will do that in order for a protein to go from one cell to another or even to different organs of the body. And that's it for translation.